Good afternoon, hello, and welcome to the Department of Biochemistry at Cambridge University in the United Kingdom. My name is Dr. Rhys Grant, and I am the Science Communication Specialist here in the Biochemistry Department, shared also with the Cell and Molecular Biology Programme at the Cancer Research UK Cambridge Centre at Cambridge University. So I'm very pleased this afternoon to welcome you to our department's second live talk as part of the brand new 2021 Cambridge Festival, which aims to bring the research of the University of Cambridge directly to you wherever you are watching this from in the world. So for this talk, I am joined by Dr. Marco Hydenan, who is a reader in protein biochemistry at Cambridge University. And today, Marco is going to be speaking to us about how X-ray crystallography can be used to determine the structures of biological molecules and how this information can then be used to facilitate the development of new drugs. I'm also joined by Latika Sugamba, who is part of our department secretariat team, and Latika is going to be helping me today behind the scenes with running this live event. Before we get on to any of that stuff though, the first thing I'd like to ask you all to please do is click the subscribe button down below so that you can follow our YouTube channel, because we have loads more talks coming up next week for the Cambridge Festival. And we're also planning on bringing you more live content from our department in the near future. I would also like to encourage everyone after today's talk to please go and check out our other social media channels on Twitter, Facebook, Instagram, and so on, using our handle at CamBiochem. So that's CamBiochem, because we use these channels to bring you all of the news, research, teaching, and information about everything really that goes on in our department. I also just want to mention that after today's talk, we'll be having a live question and answer session with Marco and myself. So if you have any questions at all on Marco's talk, or even if you'd just like to ask us more general questions on the biological sciences, if you could please post these in the live chat on YouTube, and then Marco and myself will stay for about 15 minutes after his talk to answer as many of your questions as we can get through. If you do want to participate in the Q&A session, you don't need to worry if you're sat at home in your pajamas at the moment, because you won't be seen and you won't be heard. It's just going to be me reading your questions out on your behalf anonymously. Right, so as I've mentioned, today's talk is going to be given by Dr. Marco Hydenan. So Marco began his career at the University of Helsinki in Finland, where he obtained his degree in biochemistry. He then moved to the European Molecular Biology Laboratory in Heidelberg in Germany for his PhD, which he did under the supervision of Matti Sarasti. And actually, this is also where Marco began his journey into structural biology. In 1998, Marco came to Cambridge as a postdoctoral fellow in our department, where he was working with Professor Sir Tom Blundell. He was then awarded a BBSRC David Phillips Fellowship in 2001 to start his own research group. Marco is now a reader in protein biochemistry at the University of Cambridge, and his research group studies growth factor and hormone signaling, with a major focus on structure-guided drug discovery. His research group is also involved currently in several COVID-19 research projects, and additionally, Marco has spun the company Qkine out of his research. So with that, I'd like to hand over to Marco for this afternoon's talk. So thank you very much, Marco.
Biochemistry series of Science Festival talks. What I will do in these next 30 minutes is tell you about protein crystallography, which is the method that we use in our group to study proteins, understand their function and their biology at atomic level. And I will then tell you also how this atomic understanding of protein structure will help us to design better drugs. And so the aims here for this talk is, first of all, I'll tell you a little bit about what proteins are, just in case you are not familiar with biochemistry or need a refresher on that. And I'll give you an idea how big they are, because they actually are very, very small molecules, and that's the challenge in working with them. And then I will tell you what kind of techniques we use to study their three-dimensional structure, in particular by X-ray crystallography. Tell you a little bit what those three-dimensional structures tell us, and then I will tell how we use that information for the development of new drugs, hopefully better drugs as well. But before going into the science, I just want to mention that chemistry is basically the birthplace of protein crystallography. These two gentlemen here, John Kendrew and Max Perutz, got Nobel Prize in 1962 for their studies on structures of globular proteins. They were the ones to determine the first crystal structures of myoglobin and hemoglobin, two oxygen transporting proteins that we all have in our bodies. Now, since that work by Kendrew and Perutz, there's been a massive progress in the field, and we have now nearly 200,000 structures in Protein Data Bank, which is the public repository of all the data. So everything we do in our work, when we publish it, we put into this data bank. And if you get interested in looking at these things more, it's all publicly available. Just Google Protein Data Bank or PDB, and you should be taken to the right page. We get about 10,000 new structures every year. Most of it is X-ray crystallography that I'm going to talk about today, and the rest are by NMR spectroscopy and cryo-EM, and there's going to be talks on these methods as well next week in here.
Sorry, Marco, there's something a bit weird with your slides. Would you mind stopping sharing your screen and then resharing it? There's two big grey boxes covering up parts of your...
Okay.
Which I think is something weird with the screen share.
Just a moment, I need to get back to my window here. Any better?
Close enough.
Okay, so that's far at the bottom, I take it.
That'll do.
Yeah, there's a thin line at the bottom, but
yeah, I can
see, yeah, we'll see everything else, I'm sure. Sorry for the disruption, everyone.
No worries. Okay, so like I said, we have a lot of structures available nowadays, and this really is, a foundation that we build on in our research. So let's take it back first, where these proteins come from, and this is going to be quite a human-centric approach here, but similar things apply to any living organisms in different ways. We humans are made about 40 trillion cells. It's a large number of cells: liver cells, skin cells, muscle cells, whatever part of the body you go to, it's made of cells. And each of these cells contain 2 copies of your genome. That's about 2 times 3 million nucleotides or letters of DNA code. And that's the blueprint that makes us human. And also it's a blueprint that makes us individuals because of the small differences between individuals. And that genome encodes for some 20,000 proteins. So that blueprint in it has instructions on how to make 20,000 different proteins, which are really the machines that make us work. They are the engines of our existence. And just so you, in case you have forgotten how this works, so we have that DNA, which is made of 4-letter alphabet. We call them A, T, C, and G, different chemicals which make that linear DNA molecule. That molecule, we can read that and transcribe it to a related language, which is the RNA language. It also has 4-letter alphabet. And the main difference there is that the T has been changed to U. So it's a bit like 2 different dialects of the same language, if you want to call it. But then that RNA is going to be translated into a protein. And the protein language is going to be quite different. That has 20 different letters, and it's going to sound very different from the DNA and RNA language. And those proteins, those 20 amino acids, those 20 letters, are shown here in this slide. They are different in size. You can see here in the middle, there is something called the glycine, which is a G. It's a very small one. It actually doesn't have a functional group on it. But we can go further down and there's a tryptophan or a lysine, which are much bigger amino acids. We can see from the different colors that they have different atoms in them, and that means that they have different chemical functionalities. And if you're not familiar with amino acids in biochemistry, these are the same amino acids that you take from food or you might take as a supplement if you're into bodybuilding or something. Because these are the building blocks that it needs for proteins, and your muscles, for example, are full of proteins which generate the force that moves your hands and legs. And if that was, you're not keen on chemistry as such and those molecular structures, if you remember a little bit later that this is just 20 building blocks that we have, like these kind of different shapes that you have in children's toys. We'll come back to that in a minute. Now those proteins are made into these amino acids are made into a protein by linking them into a single long polypeptide, essentially one long word in this protein language. And here is a sequence of a particular protein. We call it the CK2 alpha, and that has about 400 letters in its sequence. Now, what does that mean? So if I highlight here that short sequence PALVF on that, what does that mean? Well, on the previous slide, I showed you the chemical structures that relate to these letters in this alphabet. Now, if I look at that structure of that segment in chemistry, it looks like I show here. The little spheres are atoms, and they are connected by bonds, the sticks. I can see there is a P in the beginning there in the left. We have an A, we have an L, we have V, and we have F. Exactly the sequence that you read in here in the letters is now turned into that chemical structure. And through in the middle here, horizontally goes what we call the backbone of the protein. And that's what I've kind of just shown you here with this greenish blue line. So this is a long string of chemicals which then bundle together into a three-dimensional structure. And that's what we're going to talk about later. Now, these proteins are very complex molecules. An average protein in human genome is about 350 amino acids, and that equals 6,000 atoms. Now, that's 6,000 individual atoms that are linked together into this large molecule. Because it's a very complex thing, we want to show, we have different ways of showing it so that we can make sense of it. So here I first just show you this spaghetti view, which is just the backbone. The same line I showed you before, but now it's in this three-dimensional structure for the whole protein. But if I put all the atoms in that, it gets very busy. It's really difficult to see anything from that. Quite often we simplify that a little bit. We take the spheres away, we just show the bonds between the atoms, and now it's maybe a little bit easier, especially if we zoom into that. But we would like to make things pretty, and some 30, 40 years ago, Jane Richardson came up with the idea of showing these structures schematically, which is here with these arrows and spirals. And now it's a little bit easier to understand, especially for the trained eye. And because I've been working with this for a long time, I know that this is a protein kinase because the way it looks and how those arrows and curly-whirlies go. So you're going to see some of these representational structures later, so that's why I want to show them here. Now, what do these proteins do? Well, here's just a few examples of those 20,000 that we have. On the top here, we have an enzyme which breaks bonds. This is an enzyme called Lysozyme. We all have that in our tears, for example, because it will break bacterial walls. It helps us not to get infections by bacteria that get into our eyes. Like I'm going to talk about these signaling proteins, kinases, which is shown in the middle here. On the top right, we have insulin. This is the growth factor that regulates your glucose levels in your blood. As soon as you eat, you start secreting insulin from your pancreas, and these molecules goes everywhere in your body and make sure that cells take up that precious energy from the circulation. On the bottom, we have a transporting molecule, a hemoglobin. This is the molecule that makes your blood red. It's found in the red blood cells. Every time I breathe air in, oxygen from my lungs binds to hemoglobin. The hemoglobin in the red blood cells goes to everywhere in your body and releases that oxygen where it's needed, where your cells are breathing and doing what they need to do. And some proteins provide mechanical support. And this example of a collagen, which is a long, rigid rod, and that, for example, you find in your tissues and in your skin, which allows us to be rigid and stay upright with the help of bones instead of being like amoebas. So we have 20,000 of these, and we'd like to understand what they do at the molecular level. But the trouble is that while these are big complex molecules, we call them macromolecules as a distinction for other molecules like metabolites, which are quite small, yet they are in size quite small. So an average human being is about 1.7, 1.75 meters tall. But an average protein is about 5 nanometers. That means that a protein is 350 million times smaller than a human being. Now, those are the kind of numbers that are difficult to comprehend. So let's do a different kind of comparison. So if an average human height was equivalent to Earth's diameter, which is about 13 million meters, then an average protein would be a table tennis, a ping pong ball on Earth. So now you can get an idea on how small these proteins are to our existence. So we want to look at those at atomic level. How can we see them? What kind of magnifying glass are we going to need? Well, if I put this chemical structure here again, and don't be too scared of it, just balls and sticks, but I've highlighted some of those bonds that are holding those atoms together. So they are between 0.1 to 0.2 nanometers in length, quite small distances. Now, when I look at my screen here, I'm looking at different colors at a visible light spectrum, which is the one that my eyes can detect. And that's about a few hundred nanometers in wavelength. The distance between those peaks on that graph there. That's a lot bigger than the size of the proteins, bonds, and the distances in proteins. So it's not really useful for us when we want to look at the atomic level. So that's why we're going to use X-rays. These are the same X-rays that you would use when you take an X-ray graph of your hand or a leg, if there's a problem with it. And their wavelength is, in the nanometer range. That's why they penetrate material very well as well, and go through your, for example, hand to allow the X-ray image to be taken. So what are the ingredients then to get these structures out with this knowledge? So we're going to need some purified protein, a few samples here on the test rack. Usually they are not really colored, but here's we've given a little bit of color so you can see the liquids there. And we have some chemicals and then fancy robotics, which then hopefully, when we mix them in a particular experiment, we get these beautiful crystals. And this one here is possibly the most beautiful one we ever got in my lab. And like Reese said, my lab is doing a lot of COVID work, and this is a crystal of one of the COVID SARS-CoV-2 proteins. Protein was made by Alexi Lula, and crystallization by Paul Breyer in my lab. We're going to get those strong X-rays, and we shoot them on the crystal, and then we stick them in the computer, the data, and calculate the structure. So how does that happen in practice? Well, the experiment on getting crystals is done as follows. We're going to have a sealed chamber. It's a tiny one. I mean, it could be a very big one as well, but we want to preserve the samples. The proteins are very difficult to make. So we have a sealed chamber, and in that chamber we have at the bottom a chemical mixture, a solution of different chemicals. We call it either precipitant or mother liquor, depending on who's talking. And this is a liquid now that's going to help us to get protein to crystallize, because we're going to mix our protein and the precipitant in a single drop, a tiny drop, maybe 100 nanoliters or 1 microliter. And then we wait, and we wait, and we wait. And over the time, because we diluted the sample on that little drop, water will evaporate between these two solutions, the precipitant at the bottom and the protein at the top. And that means that it gets more concentrated, stronger solution. And hopefully, over time, we get crystallization and crystals in those drops. Okay, this is just a little cartoon for that. What happens in reality? So this is an experiment done not so long time ago, actually one of the COVID proteins again. You can see this drop here in the middle. This is about 200 nanoliters in volume. And you can see the size here. It's way less than maybe a millimeter in diameter when it starts. Next day, not much has happened. 3 days, you might think that nothing has happened, but actually there was a little thing that appeared here at 1 o'clock. Day 5, it gets bigger. Day 40, even bigger. And at 21 days, now you can see these beautiful crystals, kind of rod-shaped. But when you look from the end, they have this hexagonal structure, which probably suggests that there is a hexagonal symmetry inside. These crystals come in different sizes and shapes. The colors here are mostly artifact of the microscopes that we use. But you can see there are needle-like crystals, there are diamond-like crystals, cubes, and so on. We don't know what comes when we start the experiment, but what we hope is that these are going to be able to diffract X-rays then powerfully. Once we get the crystals, we need strong X-rays for that. And that's what we're going to get from a synchrotron. So we use mostly diamond light source. This is one of the best synchrotrons in the world. It's located close to Oxford. We used to go there ourselves and but nowadays we simply send them by. They put in them quite experienced users, and we might just get up, get on our pajamas, turn on the computer, and control the machine remotely. And here's a picture what that beamline looks like. It's a very complicated instrument. A lot of engineering goes into that. What we have here is the X-rays come somewhere from this direction. There are electrons that go around the big synchrotron ring, and we get X-rays coming tangentially from the ring. This device here in the middle is a robotic sample changer. It will put our sample into the X-ray beam, and then this detector here is a big, very expensive camera which will detect the X-rays which are diffracted by the sample. So I'm going to show you a couple of movies on what it looks like when we collect the data. So here we have this pale square here in the middle is the crystal, which is mounted into a small loop, less than a millimeter in size. We basically fish these crystals into this loop and we cool them to 100 Kelvin, minus 173 degrees of centigrade, because the X-rays are so powerful that they would destroy the crystal unless we did this. And the X-rays are going to come from here, and we're going to rotate the crystal 180 degrees because we need to image it from every direction. Just like if you might want to make a three-dimensional model of yourself, you need to take pictures of every dimension so you know how you look from the front, the side, back, and so on. So we're going to rotate the crystal here, and as we rotate it, the X-rays are going to be collected on this image here. So the crystal is turning here, and you can see we are collecting these little spots of diffraction. The problem with X-rays is that we cannot focus them. So we can have a lens that will take the picture and like in a microscope and get us the molecular structure in the end. Rather, we are collecting the diffraction, which is amplified by the fact that the proteins are in the crystal in a lattice. When I started my PhD in late 90s, it took about 24 hours to collect a full high-resolution data set. Nowadays, we get that in about a minute, and in that minute, we get 10 times more data as well. So there's been massive technological advances on that, and that's the reason why we can do, for example, drug discovery so effectively nowadays. So we take that data and we put it into a computer, and this is what we get out of it. This is what we want to see, apart from the pretty crystals, of course, because those are the ones that really tell us that the experiment might be working. So now you might look at this, and this looks, very puzzling overall. But imagine that everywhere you have this kind of a bulge on this, what we call the electron density, that's where an atom sits. And if there's a connecting density in between, that's where we're going to have a bond. So looking at that, you can see at the bottom here, we have, for example, a ring structure already. Doesn't take much imagination to see that. So let me put the atoms here. So I'm going to put atom in every bulge and every point where the direction changes here. Then I'm going to just link them with bonds where the density is connected. Now we know what the chemistry of the protein was. Remember, we had that whole sequence of the protein already known, and we can assign the individual atoms their chemical identity. Here the yellows are carbons, reds are oxygens, and blues are nitrogens. Again, I know chemistry is not the favorite for everyone. And there was the sequence, leucine, valine, phenylalanine. So if you're not keen on the chemistry, remember those building blocks I showed you before. Basically what we are doing here is we have a box. It's not holes, it's things we fill in. And we have these building blocks, and we just need to match the shape and then size to a particular piece of the density in here, just like you would do when you put these blocks into this toy. So it's just a big game for us, kind of a crystallographer Sudoku, if you want to call it. Now, what is great with crystallography is that we can not see just the protein itself, but also if other molecules are binding to that. And that's important when we're going to talk about drug discovery, because drugs are other molecules that bind to proteins. So I have here again electron density, making it a little bit more, complicated, realistic here. But just to keep it easy to see, I've colored this new density in green here. And now you can see it's a nice contour density. It's quite high quality data. But we can see that it's not linked to the blue density at all. It is simply inside the protein, but it's not linked by covalent bond. Now I can interpret those atoms again. I put them in place and put the bonds on that. And now this happens to be an ATP molecule. That's something which is really the driver of energy driver in our bodies. And it's a cofactor for this protein. And now you can see there are still kind of blobs here, which I don't have any atoms in. You can start guessing what they are. Well, they are water molecules. We are 60% water. All the proteins need water and they exist in aqueous solution. So we can actually see individual water molecules bound to the protein. And what do we do with this information? Well, this is a very complicated picture, but here we have the same ATP molecule in the middle, and I'm putting now lots of interactions, how the protein is holding the ATP together with these dotted lines. Water molecules are mediating the interactions. There are direct interactions and so on. So this really is the level of detail that we are interested in seeing in these proteins. And that will help us then with the drug discovery. So if you want to use this information for drug discovery, things that bind to proteins, we need to think what the good drug needs at the molecular level. It's a little bit different. Of course, the drug needs to cure the disease or treat the condition you have, but what does it need at the molecular level? Well, first of all, the drug needs to have a target in the body. What is the protein that is malfunctioning or something that we need to fix? So that's the kind of the starting point where we as a protein crystallographer start this journey. We need a drug that binds strongly to its target protein. That means that it has a high affinity, makes those interactions I showed, properly, good, strong interactions. It needs to have, of course, desired effect on the target, so that it prevents the harmful activity. Whatever the misbehaving protein is doing, we want to stop that. And then it should only bind to its own target protein, the one we are trying to stop from working, and not to other ones. That means that we have to have high specificity. And the reason for this is that if the drug binds to other places in your body, that might cause nasty side effects. And that means the drug is not going to be very useful as a therapeutic agent. So how can X-ray crystallography help with that? So let's take another kind of human life analogy. We can do things differently in the multiple ways of doing drug discovery. One possibility is that, as an analogy, I get an invite for a wedding, and having been sitting on in my office for the whole this COVID period and gained a few pounds, my old suit doesn't work anymore. So I need to buy a new one. So I can go to the Marks and Spencer's or Next or whatever, and they will have racks and racks of suits. And I can pick one which is, I like. But it might not be perfectly fitting. I need to tweak it a bit, shorten the legs or lengthen the sleeves or take away from the sides, whatever it might be. Eventually, hopefully, it gets into something that fits me well. But what we want to do is we want to take a made-to-measure approach, a tailored approach. So instead of going to the shop and buy something off the rack and modify that, let's measure things beforehand carefully and build the molecule is really tailor-made for our target. So one of the things that we do in particular is something kind of a cut and paste approach, which is called fragment-based drug discovery. And I'm just going to illustrate that very briefly. This is work by Paul Bray in my group and a group of David Spring, which is our kind of lead drug discovery project in my lab. So here on the left is the starting point. Some of you might have recognized that the same ATP I showed before binding to the target. And this is the one that we want to stop from going into the protein, because that will stop it from functioning. So Paul did a lot of crystallography, and he found these little fragments binding on the top part of the protein. With more crystallography, he found molecules that bind to the bottom part of this protein. And then we talked to the friendly chemist, who then become the molecular tailors, and they stitch them together. You can see the bottom part here in yellow, the top part in blue, and then this green is a linking part, which makes it into one larger molecule. And now this molecule is going to be very effective in stopping the protein from working. And this particular molecule turned out to be the most specific of this kind so far. And that's been now taken further by a biotech company locally. The other thing is the specificity. How can we improve the specificity so that the drug binds only to the target that it's meant to bind and not to other targets? Now, the problem here is that, if you remember, I told you we have 20,000 different proteins in our encoded by our genome. Lots of them are very similar to each other. And one class of proteins, which is very important for, especially for cancer treatment, are these so-called protein kinases. But we have 500 different ones in our body. They all have different roles in different cells and different biological processes. And you could think of them as close cousins. They all share common ancestors, their grandparents and great-grandparents. Some of them look very alike, some look a little bit different, but overall, they're going to be fairly similar to each other. So now I'm showing here 4 different protein kinases, all of which have drugs binding to them for cancer treatment. But if you look at this schematic view of them, you can see that they look all pretty similar. I mean, it's hard to find a difference between them in this kind of view here. So while they are very similar and it is challenging to get specific drugs, here is again where the crystallography is going to work. So I'm going to show a little bit different view of these proteins, the same 4 proteins as before. Now what I've done is they all have this binding site in the middle, a big hole. And I've cut them in half so you can see the opening, the cavity inside the protein. And you can see already the shape of the cavity is very different. The Abl kinase here has a very long curved channel. BTK maybe a little bit longer, and the SARC and EGFR have a little bit different shaped and sized cavity. The different colors here show that there are some different functionalities. Blues and reds are a little bit in different places depending on that, on the different kinase. And that just means that there are different chemical interactions we can take. Now there's an Ibrutinib, which is one of the leukemia drugs, which is binding to BTK. And you can see it fits nicely into that shape of that pocket, because with a lot of structural biology and chemistry, the pharmaceutical company that developed that, optimized the molecule for that binding site. Same for Abl kinase. Imatinib is much longer elongated molecule, and it takes advantage of that very long pocket that it has. And again, SARC and an EGFR inhibitors, the dasatinib and gefitinib, are again optimized for their pockets. And this really has been the major contribution of X-ray crystallography and structural biology on doing this rational design. Not guessing what we can do to the molecule to make it better, but look at it at atomic level and change it. And just as a final example where structural biology can help and crystallography is in tackling drug resistance. So here's the same Gleevec imatinib I showed before, a little bit different representation. And just for the simplicity here, there's a particular interaction, a bond, hydrogen bond between this threonine residue here and the drug, which is here in the middle. Now, cancer is, as we all know, a nasty disease and or set of diseases. And one particular thing with the cancer cells is that they are very good at mutating their DNA. And what happened early on when this drug was developed, that people started finding out that some cancer cells are changing, and in particular, they were changing this threonine into an isoleucine, the yellow residue here. We took the oxygen away, we put an extra atom here. Now that meant that the drug could not bind anymore, and that meant that the cancer, while it perhaps disappeared almost, it came back again. Now, what can we do to prevent that? Well, using this structural information, again, the pharmaceutical companies could develop a new drug, ponatinib, which removes the requirement of this dotted line here. And this is now a next generation leukemia drug. So as we can develop good drugs, the cancer is clever in trying to get rid of that inhibition of the drug. But again, we can play this cat and mouse game as scientists and catch these mutations, understand what they do, and overcome them with the new chemistry. So I'm going to finish here just to thank, because I'm just giving the talk here, our expertise and work is done, of course, people in the lab. Paul, Alexi, and Theo are currently in the group doing this kind of work. Gerhard, Mathieu, Max, and May were a part of the bigger team. We couldn't do this work without good chemistry collaborators, David Spring and his group, late Chris Abel, his group, and now Morten Grötli in Gothenburg. So I think with that, I finished there, and I'm happy to answer any questions you might have.
Great, thank you, Marco. That was a really, really interesting talk, and it's great to see how X-ray crystallography is being used to tackle diseases such as cancer. So it's very interesting, thank you. And so we're now going to have our live question and answer session. So if you have any questions, just post them in the live chat on YouTube, and then I'll read them out for you and we'll try and answer them. And there is a little bit of a delay between what we're saying on Zoom and you hearing it on YouTube. So I'm going to ask the first question. So I was just wondering, how long does this process of fragment-based drug discovery take? Because in my mind, I'm thinking it could be super quick because you've kind of got these Lego bricks that you're sticking together, but at the same time, it could be ridiculously long. So I was wondering how long that takes.
It depends on what stage you're talking. I mean, the whole drug discovery process is probably 5 to 10 years from beginning to end to really having a drug that you would have as a licensed medicine. But that early stage, which is the kind of getting to perhaps what we call the first in human drug, which could go to clinical stage nowadays, that could be in 2, 3, 4 years maybe done. That fragment-based approach that has become massively fast nowadays because the synchrotrons and beamlines are very powerful, and also there's a facility at Diamond called XChem where we can collect a whole crystallographic screen in just a few days. That was kind of some unheard of 10 years ago. So some of the things are very fast, but it is still, especially when you get to the stage of doing biological experiments, it gets slower because things become more unpredictable there.
Yeah, and is it essentially like playing with Lego? You are just taking bricks and sticking them together.
It pretty much is, exactly. So we have a toolbox of maybe 500 what we call the fragments, the Lego bricks, and we try them one by one, which one is fitting. And once they fit, we start looking at the different box of Lego bricks. How can we connect these together? Sometimes they are so close that, like in Lego, you put them together and link them. So we can merge the fragments, we can link the fragments, or we can grow the fragments. So there are different ways that starting point can be used.
I guess I should say that other brands of blocks are available if Lego is not the one that people fancy. We have a question on YouTube. So as new techniques such as AlphaFold, which determine protein structures computationally emerge, do you think it would become an alternative to X-ray crystallography? I'm guessing this is a scientist that's asked this question.
Yes, just maybe background to other people. There was a Google-funded program called AlphaFold, took part in a big kind of scientific competition on predicting computationally, trying to get, make us unemployed as crystallographers. And they were doing a very good job at that. So eventually, I think, yes, I think there's still probably too many uncertainties in predicting binding of, for example, these fragments onto even experimentally defined structures, let alone predicted structures. But, it'll be coming for sure. Hopefully not too soon, a few years before I retire.
I guess it's a sort of linked question. So we have talks coming up next week on cryo-electron microscopy and nuclear magnetic resonance spectroscopy. So why is X-ray crystallography the best, in your opinion?
It's not
it's going to start a war with other researchers.
Yes, so there's a lot of course, competition between methodologies. It's not the best in overall. It depends on what you need to do. The best technique is what answers your question best. At the moment, crystallography in throughput, once you have the crystals, and that can be difficult, but once you have crystals where we can actually, because they actually have big channels of water in them, so we can put them in solution of a chemical, and chemical goes inside. So we call it soaking, like a sponge will soak a liquid. If we can do that, then we can get in a single day 100 structures. And that's simply orders of magnitude more than we could get by NMR or cryo-electron microscopy. But there are proteins that we cannot study by crystallography. And then the other techniques are going to be more useful. Cryo-EM is going to get faster for sure. So and then improve. So that's another thing that's going to make us possibly unemployed one day.
Is the reason
make us learn new things, let's put it this way.
Is the reason that some proteins can't be studied by X-ray crystallography because they won't crystallize, or? Well,
the crystallization is the biggest hurdle there. We don't really have a size limit. We can crystallize, even whole viruses can be crystallized. So very large structures can be crystallized in the molecular terms, large structures. But there are lots of proteins which don't have a defined three-dimensional structure in solution. Those are unlike the ones I showed. And those, for example, are the ones that we will study by NMR spectroscopies. Again, very large, some very large complexes are too difficult to make in the quantities we need for that. And then the cryo-electron microscopy has already a significant advantage.
Yeah. Latika, do you have a question that you wanted to ask, or?
Not really. It's more, I guess it's a bit silly. I just think that the pictures that you got are really, really cool. Is there a way that you'd be able to provide some of those just on your website or something? Because they'd make really cool backgrounds.
Well, we have some of those in there. I mean, that's part of the fun of doing crystallography. It's quite visual. First of all, the crystals are beautiful, and the electron density, as such, is quite nice to look at, and it is that puzzle element of that. But a lot of it is about making those. It's a complex set of data. We have lots of atoms there, so many of those images that I show, they are really simplified views of what goes on in there, because there is too much information. It's visually too busy. I mean, we have on our group website pictures of those, and if anything I have on my talk, happy to share. No problem.
Awesome. Yeah, I think it's something that I'll be maybe copying and putting into some of my artwork.
Yeah. Definitely not a silly question. They are beautiful images.
They are really cool.
And actually, what determines the shape of these crystals? Because you had some long needle-like ones and some that were more like sort of what we kind of picture
different things.
looking
I mean, it's a short talk, so I couldn't go, but the crystals will have certain symmetry inside them. 2-fold symmetry, 4-fold symmetry. They are packing the molecules differently in there, so that has some reflection on the shape. So the ones I showed, which had a kind of a hexagonal end, they probably have a hexagonal symmetry inside. So 6-fold symmetry. So 6 molecules which are rotated 60 degrees each. What governs the outer shape, like the long needle, is that the proteins are growing. Proteins need to stick to each other to form the crystal. And if they're growing very fast in one direction compared to sideways, lengthways very fast and slow in sideways, the crystal will grow lengthwise. If they all grow in an equal speed in all directions, then they become more three-dimensional and rounded, or a cubic, or whatever it might be. So it's again completely unpredictable and difficult to control.
They are beautiful, beautiful images. And someone actually has just put in the chat a message for Latika saying that you should search for the PDB screensavers.
I will definitely do that.
Database or protein data bank. Yeah, they've got screensavers available. We're actually 5 minutes over our session, so I think I'm going to wrap this up now. So I just want to say thank you very much, Marco, for a very interesting and beautiful talk. Thank you very much also, Latika, for helping me with running this afternoon's event. And I just want to also say thank you very much to everyone at home for watching the talk and for joining us this afternoon. And we have loads more talks coming up next week for the Cambridge Festival. So do subscribe to our YouTube channel, and hopefully we'll see you again soon. Stay safe and have a lovely weekend.